sincerely pray you're well, holding fast, standing strong in faith. I was led to add this bit of information regarding actual human history according to the Bible. We certainly are in the days of Noah, but the sons of God no longer take wives unto themselves to mingle themselves with the seed of men, because by their sorceries were all the nations deceived.
There were giants in the earth in those days, and also after that, when the sons of God came in unto the daughters of men, and they bare children to them. The same became mighty men, which were of old, men of renown. And God saw that the wickedness of man was great in the earth, and that every imagination of the thoughts of his heart was only evil continually. And it repented the Lord that he had made man on the earth, and it grieved him at his heart. And the Lord said, I will destroy man whom I have created from the face of the earth, both man and beast, and the creeping thing, and the fowls of the air, for it repenteth me that I have made them. But Noah found grace in the eyes of the Lord. These are the generations of Noah. Noah was a just man, and perfect in his generations, and Noah walked with God.
If we are software-driven machines like every other biological species on this planet, understanding how to interpret that software, including the parts of the software associated with interacting with the environment and feedback control mechanisms, we will start to understand our species quantitatively for the first time. Everybody's asked questions, what's nature and what's nurture? We will be able to solve in the next decade mostly of what's nature. By difference, everything else is nurture.
John Craig Venter, an American biotechnologist and businessman. He's known for leading the first draft sequence of the human genome and assembled the first team to, now make note of this, transfect a cell with a synthetic chromosome. Transfect a biological, naturally occurring cell like a human cell with a synthetic chromosome. Think about that. John Craig Venter, October 14th, 1946.
So how do we boot up a synthetic chromosome? So we had teams working in parallel on this. This, I think, is one of the most profound studies we published, whereby swapping out the genetic code in a cell, we completely converted one species into another. We completely converted one species into another. We completely converted one species into another. And because this is so important, let me walk you through that. So there's two mycoplasma species that we use. M. mycoides, we isolated the chromosome from it. We treated it harshly to remove any proteins, because for synthesis, we wanted to prove you could just work with naked DNA. We added a couple extra genes to that chromosome so it would turn cells bright blue, and so we could select for it. And we injected this into a second mycoplasma species, capricolum. These are about the same distance apart as human and mice in terms of a genetic code. So we injected the new chromosome into the cell, and now briefly we have a capricolum cell with two sets of instructions. As with the viral DNA, the new DNA started to be read, and some of the early proteins produced were restriction enzymes. This is what my colleague Ham Smith discovered and got the Nobel Prize for in 1978. That's allowed this whole field to go forward. We think these enzymes recognize the parent chromosome in the cell as foreign DNA and shoot it up. Recognize the parent chromosome in the cell as foreign DNA and shoot it up. So now we have a cell of one species, a cytoplasm of the capricolum, and the genetic instructions from mycoides. In a very short while, we had these bright blue cells, and when we interrogated them, all the caprcolon proteins were gone, replacing completely everything with the new, driven by the new genetic instructions that made mycoides proteins. So simply by swapping out the software, we converted one species into another. So we use these new synthesis techniques to go back and synthesize the complete chromosome very quickly, just starting with the digital code in the computer. So this is the first cell that actually has as its parent, its genetic parent, the computer. So this is the first cell that actually has as its parent, its genetic parent, the computer. And to prove this, because we wanted to make sure we couldn't get into troubles with contamination of a single molecule of DNA, we've learned various ways to watermark the DNA, inserting things in the genetic code. Is we had a code within the genetic code that gave another code. And if you decoded that, you could actually read in the English language various things that we wrote in the genetic code, including the names of 46 scientists that contributed to this working, the names of 46 scientists that contributed to this working.
So, in effect, these 46 scientists now own this new species. Do you understand?
And whereas thou sawest iron mixed with miry clay, they shall mingle themselves with the seed of men, but they shall not cleave one to another, even as iron is not mixed with clay.
I made this video short so you could go back and fully wrap your mind around what's going on here. And don't forget, this research you're watching right here goes back 13 years.
This, I think, is one of the most profound studies we published, whereby swapping out the genetic code in a cell, we completely converted one species into another.
So you can only imagine what other advancements have been made in that 13-year time. And when you do realize what's actually going on, call on the name of Jesus. God bless each and every one of you. Peace and grace to you. Many fish.
Finally, my brethren, be strong in the Lord, and in the power of his might. Put on the whole armor of God, that ye may be able to stand against the wiles of the devil. For we wrestle not against flesh and blood, but against principalities, against powers, against the rulers of the darkness of this world, against spiritual wickedness in high places. Wherefore take unto you the whole armor of God,